Hey guys, so this lesson is going to be going over mitosis. Now, way back in grade 9, you guys should have learned about mitosis, but I realize that's been a while, so we're going to review it fairly in depth now. So we are going to talk about what the purpose of mitosis is and what you end up with at the end of mitosis. You also need to know each of the phases of mitosis, including the order of the phases, as well as what happens in each phase. And then at the end of all of those different phases of mitosis, we have cytokinesis. So you need to know what that is as well. So mitosis is the way that somatic cells divide. So just a reminder, the somatic cells are all of the cells of your body, with the exception of your sex cells, which don't act like the rest of your somatic cells. Mitosis is the way that your genetic information divides, so your DNA essentially. And at the end of mitosis, you will end up with 2 new diploid cells. And just a reminder, by diploid, we mean that they have 2 copies of every chromosome. Each of those 2 new cells will be identical to each other. They will also be identical to the parent cell or the original cell. So we call the new cells the 2 daughter cells and the original the parent cell. In eukaryotes, like ourselves, that is any organism that has a nucleus, this process is used for growth and repair. For organisms that reproduce asexually, mitosis is also the way that some of them reproduce. Mitosis is the division part of the cell cycle, as well as cytokinesis, which will happen immediately after mitosis. And it's made up of 4 phases: prophase, metaphase, anaphase, and telophase. Prophase is what happens right after interphase. So in interphase, you have the chromatin, which is the uncondensed version of our chromosomes. And then at the start of prophase, that chromatin will condense into the actual chromosomes that we see. The chromosomes will be double-stranded in prophase because if you remember, during the S part of interphase, the chromosomes replicate. So they go from single to double-stranded during the S portion of interphase. And so by the time you get to prophase, all of the chromosomes are already double-stranded. So there'll be that X shape. During prophase, you'll also see the nuclear membrane start to disappear, as well as the nucleolus, which is like a little dark area inside of the nucleus. There's also organelles called centrioles in the cytoplasm. Normally they hang out as like a pair together, but during prophase, they migrate so that they're on opposite ends or opposite poles of the cell. Those will now be on the 2 different sides. You'll then get spindle fibers start to form from the microtubules in the cytoplasm. They'll form what's called a spindle apparatus. They're going to connect to the centrioles that are on the opposite sides of the cell, and then they'll also start to connect to the centromeres of the chromosomes. So the centromeres are the things that hold the 2 sister chromatids together. By the way, plant cells don't have centrioles, so this only happens in animal cells like this, but plant cells do still make spindle fibers and the spindle apparatus. So this is kind of what that looks like. So here in this top diagram, we have like the very, very start of prophase. So the nuclear envelope, or also called nuclear membrane, is starting to break apart. The chromatin at this point is still uncondensed, so it still is chromatin. And our little centrioles are still close together. And then what'll happen is, as we actually move away from interphase into prophase, you'll have the chromatin condense into these X-shaped chromosomes. The centrioles will go to opposite sides of the cell, and you'll have those spindle fibers that are coming off of them. And the nuclear membrane, again, is breaking down. Then we move into metaphase. So the spindle fibers are attached to the centrioles, and they're also going to attach to the centromeres, the middle part of your X-shaped chromosomes. And these spindle fibers will pull on the chromosomes until it causes the chromosomes to line up in a straight line across the middle of your cell. The middle of the cell is sometimes referred to as the equatorial plate, the equator, or the metaphase plate. It just means across the middle of the cell. And the cell will sometimes start to elongate a little bit at this point. So that's what we can see at the top here is kind of our line of chromosomes. And then on either side, we have our centrioles. These are our spindle fibers. In anaphase, the spindle fibers start to retract or get shorter. And essentially what they're doing is they're like reeling in their chromosomes. So you have that kind of like X-shaped chromosome in the middle. And then as it gets pulled apart, they're going to get pulled, the sister chromatids are going to get pulled apart from each other and pulled to opposite sides of the cell. The chromosomes do form like a V shape as they're pulled, because the chromosomes will be like hanging out like this, kind of that like X shape. And then when they get pulled, as they're pulled through the kind of like thick jelly, they like bend as they're pulled. It's like if you had a piece of string and you grabbed it by the middle and pulled on it, it would also form kind of like a V shape. So yeah, so what's gonna happen is then all of like the right side sister chromatids get pulled to the right side of the cell, all the left side sister chromatids get pulled to the left side of the cell, and then you will have a group on either side of the cell, and those two groups will still be identical to each other because sister chromatids are identical to each other. So yeah, each hole of the cell will contain all of the DNA that has ever been present. We're just essentially going from double-stranded to single-stranded DNA. Yeah, this is kind of as we move from metaphase to anaphase. The kind of cartoon version here is the cells, the chromosomes all lined up across the center of the cell and then getting pulled to opposite sides as these sister chromatids separate. If you were to look at this under a microscope, a fairly kind of straight dark line like this in the middle of the cell would be metaphase, and then two lines, and if you zoomed in on close to that, you'd see like little tiny V shapes. That would be your anaphase. And then telophase is the last phase of mitosis. These spindle fibers will dissolve. You'll have nuclear membranes reform around the like groups of single-stranded chromosomes that are now on either pole. So each nucleus will reform around 46 chromosomes. When they were lined up across the middle, you had 46 double-stranded chromosomes. Now you have two groups of 46 single-stranded chromosomes. And then the chromosomes at this point will start to lengthen, they'll start to uncondense and turn back into chromatin. And then you'll also have this pinching in of the cell membrane that you see at the top here, sometimes referred to as cleavage. And that's getting us ready for the next part, which will be cytokinesis. So this is just kind of a summary. We've got interphase at the beginning here, where our nucleus is surrounding the chromatin, the uncondensed genetic material. Then in prophase, the centrioles have moved to opposite sides of the cell where they used to be close together. Our chromatin has condensed into actual chromosomes, and our nuclear membrane has disappeared. In metaphase, you have the lining up of the chromosomes across the equatorial plate or the middle of the cell. In anaphase, you have the sister chromatids pulled apart and pulled to opposite sides of the cell. And then in telophase, you have the nuclear membrane reforming around those groups of now single-stranded chromosomes and the pinching in of the membrane. And then cytokinesis will happen right after telophase. So once the genetic material of the cell has been divided into two, then the rest of the cell has to divide. So this is just when the organelles and the cytoplasm and all that kind of stuff is split between what is going to be the two new daughter cells. And then you will have that the pinching in that's happening between the two new cells. That'll just keep deepening and deepening. That's called the cleavage furrow, is that kind of deep V that's going to form until eventually those two sides that are like pinching in will just touch, and then the two cells will separate from each other. In plant cells, the cell wall is too sturdy for that to actually happen. So you don't have any of that pinching in or cleavage happening in plant cells. Instead, you have something called a cell plate, which is kind of like a double thick cell wall that is formed across the middle of the plant cell to separate the two halves. And then eventually that cell plate just becomes the cell walls of those two plants. So this is what it would look like. At the top here, we have telophase, and then this cleavage will just keep pinching in more and more until those two sides come together and the two daughter cells separate. At the bottom here, you can see what it looks like in plant cells. So you just have this cell plate form across the middle, and then that will develop into the two cell walls. And this is what it would look like under a microscope. You can see the kind of pinching in happening. There's not complete separation. There's still a little bit of connected area between these at the moment, but that is almost done with cytokinesis. And here in the plant cell, you can see this line that's forming. That's the formation of these cell plates that's happening. Okay, so that's it. Later on in this unit, we're going to be talking about meiosis as well. And then following that, we'll kind of do a big comparison of meiosis versus mitosis.